In this video, we are going to talk about the one-step multiplication curve of viruses. And a multiplication curve is just showing how the virus population is multiplying over time. So here are kind of the steps in this multiplication curve. And so initially, check out these axes. We're looking at the number of infectious virions. Remember, virions are intact virus particles capable of infecting a host. And then we're looking at time, okay, across the x-axis in hours. So what we're looking at first, I mean, a lot of times this is done in like in the lab. And if we want to kind of see how long it takes for these, replication cycles and things to happen. And so initially, there's going to be inoculation. So you're introducing, there's my words, introducing virus particles to a host cell. So at this point, initially, right off the bat, we might have, hundreds of virus particles that are intact, ready to find a host. Well, notice that this number decreases and eventually, I don't know if plateaus isn't the right word, but like it levels out. And that's because as virus particles start infecting or start attaching to a host, right, they uncoat, right? Once they get into the host, they fall apart. So we no longer have intact virus particles, right? So this number is decreasing as the viruses are entering the host. And then the eclipse is where there are no intact viruses because the penetration stage has happened, okay? And then eventually they're going to go through biosynthesis and maturation, and then they're going to start exiting the host cell, right? And so this is why the number of virus particles are drastically and rapidly increasing, because at this point, many virus particles are exiting from a single infected host, okay? And so this is going to continue to happen until finally all the virions have been released for each bacterium. So we refer to burst as the process of the virus particles bursting out of the host. And then burst size is how many virions are released per bacterium. That in the population or in the environment, right, there would be lots of viruses being released from lots of different host cells. All right, so this is our multiplication curve. Now, we can categorize animal virus infections kind of just based on their behavior and their replication cycle. So acute infections come on quick and they go away quick as a viral infection can go away. So this is kind of where, you go to bed one night, everything seems fine, then you wake up and you just feel like you've been like hit by a truck, right? So this rapid onset of symptoms, and then because it's a viral infection, there aren't antibiotics or anything like that. So you have to wait it out, and eventually your immune system is going to clear it, and then it's done. Okay, so normally short for a viral infection is like 10 to 14 days. So persistent infections are where the virus hangs around for a long time, months, years, sometimes even a lifetime, okay? So notice either intact viruses or their genetic material are present in the host, okay? And the person may or may not have symptoms, kind of depends on the virus and kind of what it's doing inside of the host, okay? But again, this can continue for a very long time. Latent infections are going to be where there's a delay between the infection by the virus and those lytic events where the host cells start to die. So a lot of times this is where there's a provirus involved, so HIV, herpes, okay? And so the provirus can be integrated into the host chromosome. In some cases, it can replicate separately, kind of like a plasmid. But this is when the virus cannot be eliminated. There is no cure. It is with you for life, okay? And so what happens is the virus can remain inactive for long periods of time. So whenever somebody has a cold sore, that's from a herpes virus. It's HSV1, and it's oral herpes. And people get it from coming into contact with the virus from a cold sore. And so it's often, I think it's like 85% of the population actually has oral herpes. Not everybody gets cold sores. Again, it depends on the host. But that's why people don't have cold sores all the time, because when the virus is inactive, that means it is not actively replicating. There's no signs or symptoms, right? But a lot of times when the host is under a lot of stress, that stresses out the virus reactivates, it begins going through that replication cycle, host cells start to die, and then that's when the signs and symptoms may appear, for instance, like a cold sore, okay? And just FYI, because it is so common in the population, a lot of people wonder how they get it. And a lot of times, little kids actually come into contact with the virus because, let's say, for instance, someone's dad gets a cold sore and the virus is present in that cold sore. And so if the dad is drinking out of a cup, right, and sets it down, and a little kid comes up, grabs the cup, doesn't know any better, starts drinking from it, that's how the virus may then have gone from the dad's lip onto the cup, and then from the cup onto the kid. Or maybe the kid kisses, the dad and comes into contact with the virus. So often the kid oral herpes is acquired as a kid because they just don't know any better. They don't know to avoid, contact with anything that the cold sore has come into contact with. So we'll talk more about that when we get to Unit 4, but it's something a lot of students say, oh, I have cold sores, and, I never even knew how I got it. And so it's because a lot of times it's as an infant or a toddler or something. Okay. All right. Let's go ahead and think about oral herpes, like I just said. So I kind of gave it away in this video, but that's okay. So Jody has oral herpes. Whenever Jody gets really stressed out, she gets a cold sore on her mouth, but she doesn't always have cold sores. Herpes viruses stay with the host for life. What type of infection is this? So that would be a latent infection, because again, when the virus is inactive, there's no cold sore. When they're stressed out, when the host is stressed, the virus is stressed, and then it starts replicating, and that leads to the cold sores. So this would be an example of a latent infection. All right. Now there's one last thing about viruses that I want to mention. It kind of fits in this conversation, and that's how viruses are studied or replicated in the lab. It's a lot harder to grow viruses, not grow, that's the wrong term. Viruses aren't alive. Cultivate viruses in the lab because viruses require living hosts. So unlike bacteria, right, we grow those on an agar plate. It's just nutrients and agar, and they grow all by themselves. Viruses aren't that easy because of that living host requirement. And so a lot more time, energy, money, resources go into cultivating viruses in the lab. And some viruses are so specific that it's darn near impossible to even cultivate them in the lab. So it takes a lot to study viruses and to really make a lot of progress when studying viruses. It's definitely a lot harder than a lot of like bacterial pathogens and other pathogens that we'll talk about. All right, y'all, that is the end of our conversation on viruses. Let me know if you have any questions.